Hello, my name's Aiden Willis, and today I'm going to be presenting ectopic volar fibroblasts induce histologic changes in non-volar skin. This is research that I participated in as part of the Garza Laboratory within the Dermatology Department at the Johns Hopkins School of Medicine. I conducted this research under the direction of my PI, Dr. Luis Garza, and my lab manager, Dr. Sam Lee. I would like to briefly give some background information before I actually get into the results of this research. It's known that some skin possesses unique functional adaptations that optimize it for certain scenarios. For example, volar skin, which is found on the soles of your feet and the palms of your hand, is particularly resistant to pressure and shear due to an increase in epidermal thickness, and this increase is driven by a thicker stratum corneum and increased collagen fiber density. Conversely, non-volar skin lacks this functional adaptation and actually is more prone to degradation when exposed to pressure and shear. Our lab, this project was conducted as part of the STUMP project within the Garza Laboratory, and this project seeks to alter skin identity at amputation sites to improve quality of life for amputees. Typically, non-volar skin is used to graft amputation sites. However, this skin is prone to chafing and degrading when exposed to a prosthetic for an extended period of time. Therefore, by changing skin characteristics so that they're more like volar skin, we can improve quality of life by preventing this degradation. Our research focused primarily on fibroblasts. And fibroblasts are the cells within our skin that produce collagen fibers. And as I stated earlier, volar skin is characterized by increases in collagen fiber density. It's also known that fibroblasts retain positional identity when transplanted and engrafted. And by doing so, it is possible for these fibroblasts to signal these cells around them as they would in their original location. Therefore, we seek to alter skin identity by transplanting volar fibroblasts into non-volar locations to produce increased epidermal thickness. To do this, isolated fibroblasts from sole and scalp biopsies were amplified and injected into volunteers. And after 2 or 5 months, the injection tissue was excised for further examination, and we quantified the effect of injected volar fibroblasts by measuring epidermal thickness, keratinocyte cytoplasmic size, collagen fibril length, and keratin 9 expression. As I stated before, volar skin is characterized by increases in collagen fiber length, or increases in collagen fiber density. Therefore, it would be expected that increases in collagen fiber length would result in an increase in epidermal thickness. This is actually what we observed. We observed that injecting volar fibroblasts into non-volar skin did result in increased collagen fiber length. Further, we focused on keratin 9 protein and mRNA expression because previous research has shown that the volar epidermis uniquely expresses keratin 9. Therefore, if we observe keratin 9 protein and mRNA in a non-volar site that was injected with volar fibroblasts, this would provide evidence that the volar fibroblasts are in fact signaling surrounding cells and altering protein and mRNA expression to produce skin more similar to that observed in volar areas. Keratinocyte cytoplasmic size is important because this is a further evidence to suggest that fibroblasts are signaling surrounding cells to produce thicker epidermis. And finally, epidermal thickness is the culmination of the previous 3 variables. You would expect to find injected volar fibroblasts would increase epidermal thickness when compared to normal skin treated with a control. And what we observed was that across the board, across all variables, injection of volar fibroblasts into non-volar skin produced statistically significant increases in epidermal thickness, keratinocyte cytoplasmic size, keratin 9 protein expression, and collagen fiber length. So in conclusion, the purpose of the study was to demonstrate that injection of volar fibroblasts into non-volar skin can drive changes in the characteristics of this non-volar skin, defined by increases in epidermal thickness, keratinocyte cytoplasmic size, collagen fibril length, and keratin 9 expression. We observed statistically significant increases across all these variables, which suggests that injection of volar fibroblasts allows these fibroblasts to retain their original positional information and can alter the characteristics of non-volar tissue when transplanted to a new location. Thank you for your time today.